Okay, so my chosen research article was primary cilia on muscle stem cells are critical to maintain regenerative capacity and are lost during aging. So basically the study description, the main research question that they're asking is going to be how does primary cilia on the muscle stem cells affect regeneration capacity? And this is going to occur specifically during aging is what they're looking at. Their overall goal was to find greater info on the signaling dysfunction in aged muscle stem cells. They also wanted to find out whether the ciliary hedgehog, which is also known as the capital H lowercase h signaling pathway, can be used as a potential therapeutic target in muscle regeneration. And then they also wanted to find the different specific ways in which primary cilia is lost during aging. So figure 2 is going to focus on the loss of primary cilia and muscle stem cells reduces the proliferative and self-renewal capacity. So the research question here is going to be, does loss of primary cilia and muscle stem cells affect proliferative and self-renewal capacity? So we have the experimental design and methodology. So first they started by elucidating the role of primary cilia with the RNA sequence analysis. So that's what you're going to see here at 2A up here. This was shown that the specific pathways were expressed differently. This was in accordance with the known cilia. And so the control is just going to be the muscle stem cells, and then the experimental is the IFT88 -- So they then confirmed this using a qPCR, which we see here. The control showed a greater capacity than that of the experimental groups. Then this figure 2 continued. They further confirmed using an EDU incorporation assay. That's what we see here at D. The knockdown here showed similar components needed for cilia assembly, and this confirmed the primary cilia role. They then plated the control and the IFT MUSCS on 2 collagen-coated plates. Here we see that at 2E. Basically, they did not differ from the ciliated controls in differentiation. Both showed the ability to form MYHC plus positive myotubes. This was consistent in the hypothesis that primary cilia mediate the muscle stem cells proliferation and expansion. So then there was an engraftment study. Basically, the control and experimental were transduced with a GFP luciferase expression vector, and this showed that the experimental engraftment was reduced, and that just basically meant that the cilia are indeed crucial to self-renewal. So the results of figure 2 are basically, so the RNA sequence showed that specific pathways were found to be expressed differently in accordance with known cilia function. The PCR furthered that and control showed a greater capacity than that of the experimental groups. The EDU incorporation assay basically showed that the knockdown showed similar components needed for cilia assembly. This confirmed the primary cilia's role. Then the plated controls and the experimentals, they did not differ from the ciliated controls, the experimental ones didn't, in differentiation. So basically they both showed the ability to form MYHC positive myotubes. This was consistent in our hypothesis that primary cilia mediate muscle stem cell proliferation and expansion. And the engraftment study found that the experimental engraftment was reduced and cilia are crucial to self-renewal. So then we have figure 4, and basically the focus here is that aged muscle stem cells present decreased ciliation and hedgehog signaling. The research question asked here is, do aged muscle stem cells harbor a ciliation defect that contributes to their loss of stemness and regenerative capacity? Basically what they did here is they used the immunofluorescence analysis. It showed that the aged muscle stem cells had significant decrease in both the long and short cilia, versus the young did not. Then both the negative regulator and the downstream effector were just regulated in the muscle stem cells while they were not in the young. They then did a injury time course, and basically during this, the aged muscle stem cells had an increased negative regulator, which is the PTCH1, and was seen at this increased negative regulator expression was seen at uninjured day 1, 3, and 16 post-injury. Meanwhile, the young muscle stem cells, the negative regulator was induced in injury response. In day 3 post-injury, the young muscle stem cells, the downstream effector, also known as the GLI2, was transiently upregulated. I'm sorry. And in the old muscle stem cells, the downstream effector showed decreased expression. So the basic results were that a cell intrinsic ciliation defect in aged muscle stem cells caused a numb response to hedgehog signaling versus that of the young who responded normally to the hedgehog signaling. The conclusions from this experiment were that the self-renewal and regenerative capacity in adult muscle stem cells is dramatically decreased with age due to genetic ablation. And then loss of ciliation in aging is caused by an intrinsic defect of muscle stem cells. This can be combated by activation of the hedgehog pathway. Future questions would definitely be that how could this be used to treat muscular injuries in athletics? How could this be used to slow the aging in skin? And personally, the one that I thought would be very interesting is, could we further this experiment into the aging process of eggs in females? So looking at, the age frame for women to give birth and how could we look into that? This is my citation. And that's it. Thank you.